Welcome back, everyone. Our third session is titled Advances in DMD. It will focus on the latest research and innovations in DMD treatment. Our chairpersons are Professor Dr. Ichizo Nishino, the Director of the Neuromuscular Research at the National Institute of Neuroscience in Tokyo, Japan, Professor Dr. Ahmed Iheb, Professor of Human Genetics at the School of Medicine at NGU, and Professor Dr. Kristen Schaff, Managing Director at the Institute of Human Genetics at Heidelberg, Germany.
So first, I would like to welcome Dr. Iman Rifai. Dr. Iman is a medical scientist. She's working in translational research in neuromuscular disorders. She had her bachelor degree in medicine and general surgery from Tanta University and completing her PhD in medical biochemistry from Osaka University. She's currently working as a trainee at the National Cerebral and Cardiovascular Center in Osaka, Japan. And also she's working in research. Her research is dedicated in understanding the treatment of neuromuscular disorders, pathogenesis and treatment, and bridging the gap between laboratory and clinical application. Go ahead, Dr. Iman.
Thanks so much for the introduction, and good afternoon, everybody. Today, I'd like to present my PhD project about LSMEM2 protein. This project was held in Japan, in Osaka University, in cooperation with National Cerebral and Cardiovascular Center. These are today's outline I'm going to cover. LSMEM2 protein is a leucine-rich single-pass membrane protein, an orphan protein with an unknown function. The Human Protein Atlas shows that LSMEM2 protein is expressed at heart muscle and skeletal muscle. LSMEM2 protein has two main domains, intracellular N-terminal and extracellular C-terminal, with a transmembrane portion which is leucine-rich. LSMEM2 is localized at chromosome 3 and it has 4 exons, and these exons are translated to LSMEM2 protein 164 amino acid. Previous report shows that LSMEM2 bind to alpha-2 domain of AMBK, and in vitro study at C2-C12 medium shows that part of LSM2 is co-localized with AMBK alpha-2 in the perinuclear region. Previous studies has been conducted at our labo in Japan as the heart study, and we were able to localize LSMEM2 at the heart intercalated disc with cardiomyocyte specific expression pattern. In the heart study, we found that the intracellular N-terminus has serine 37 residue, which is phosphorylated with AMBK. So we thought that the C deletion mutant is a constitutive active mutant and is a gain of function mutant. That's why we produced a transgenic mice model of the heart, and we found impairment of the ventricular function at C deletion mutant in comparison to control and wild type overexpression. And this pathogenesis could be related to inflammatory infiltration that had occurred at the left ventricle. The purpose of my study was to demonstrate the subcellular localization and function of LSMEM2 in skeletal muscle. Droplet digital PCR shows that LSMEM2 is expressed in the heart and skeletal muscles with type 1 fibers and type 2 glycolytic fibers, tibialis anterior, EDL, soleus, gastrocnemius, and quadriceps. We generated a knockout model using CRISPR-Cas9 gene editing, which is a loss of function model. And this model was successfully showed successfully knockout of LSMEM2 protein, as demonstrated here at the Western plot. Interestingly, when I held the immunohistochemistry, I found LSMEM2 protein is localized at the neuromuscular junction, and the signal merged with bongratoxin, which is a marker for acetylcholine receptors. And moreover, LSMEM2 knockout mice were healthy and fertile, and no histopathological or functional abnormalities were detected. Then we were able to produce a gain-of-function mutant using AAV constructs as overexpression models for wild type and C deletion mutant. And we used the BA tag as a BA and the flag tag markers for this construct. Why we choose the C deletion mutant? Because as I mentioned before, C deletion mutant, we think that it is constitutive active and a gain-of-function mutant. Then we introduced LSMEM2 wild type and CDEL mutant and MOC as a negative control into the mice. It were intramuscularly injected at the age of 8 weeks, and after 8 weeks we harvest and did other investigations. And also in the overexpression model, we found that LSMEM2 is expressed at the neuromuscular junction, as similar to the endogenous expression. However, at the C deletion mutant, we observed accumulation, or we can say change of localization or additional localization as the form of intracellular vesicles, which appeared at C deletion mutant. And these vesicles appeared in both oxidative and glycolytic fibers. Interestingly, in C deletion and wild type overexpression models, we observed nuclear localization, and this localization has not been confirmed in endogenous samples yet, but it has been present in the overexpression model. In the overexpression models, we also found, or there has been appeared tubular aggregates which were confirmed in modified Gomery stain as the wild type and CDEL mutant. These tubular aggregates are sarcoplasmic reticulum derived tubular structure, and we observed it with electron microscope, and they only appeared as the wild type and CDEL, while it never appeared as the MOC injected mice. Accumulation of this tubular aggregate is impairment of protein clearance, which is found in two types of muscle pathology model: congenital myopathies and male aging mice. We conducted NAD-HTR staining to find if these aggregates is positive for NAD-HTR staining or not, and they were negative, which mimic the aged pattern of the tubular aggregates. Moreover, in the female injected mice, the data was reproduced, and we found LSM2 localized at the neuromuscular junction, and also the intracellular vesicles appeared in the C deletion mutant. However, in modified Gomery stain, there was no tubular aggregates appeared. And from previous reports, we think that these tubular aggregates never appear in female mice. It only appears in male mice. To measure the force in situ, we use the Aurora scientific machine as you see here. And I also shared the QR code at which you can see the detailed methodology for how we measured the muscle function. And interestingly, we found that the twitch force, tetanic force, and force frequency were impaired at the C deletion mutant. However, the muscle weight was not affected. So we think that muscle dysfunction not caused by atrophy or tissue loss. Then I confirmed RNA-seq analysis using gene set enrichment analysis software, and I found that there was a common pathways affected in both male and female. These pathways were mitochondrial translation, mitochondrial gene expression, mitochondrial respiratory chain complex, ER to Golgi vesicle mediated transport, and oxidative phosphorylation. However, I also found that the unfolded protein response was significantly affected in male only, but not in female. Maybe this could explain why we found the tubular aggregate only in male mice, not in female mice. And to confirm our results of the RNA-seq analysis, I confirmed immune histochemistry staining using mitochondrial marker, COX-1 and ATP5F1. And I observed peripheral accumulation of mitochondrial signal at the periphery rather than the central of the myofibers, which was significantly detected at seed deletion mutant. Using line scan analysis with image software, I was able to quantify this data. Moreover, this finding was confirmed with transmission electron microscope results, and we found that there was subcircular mitochondrial accumulation at seed deletion mutant, which not appear at the mock control. Moreover, these data were reproduced in female using COX-1 and also ATP5F1. The peripheral accumulation of mitochondria was increased in seed deletion mutant rather than the mock control. Furthermore, we want to identify these intracellular vesicles. So I used the endosomal markers for these vesicles as REP5, which is an early endosomal marker, and I found that these intracellular vesicles merged with REP5 as a seed deletion mutant. Moreover, I used another marker for endosomal, which was LAMB2 marker, and also these vesicles merged with the LAMB2 marker. And these data were reproduced in female samples as well. These intracellular vesicles merged with REP5, and also merged with LAMB2. Then I want to talk about laminopathy. What's laminopathy or nuclear envelopathies? Laminopathies are a group of disorders caused by mutation in lamina gene encoding A-type lamines, which is a component of the nuclear lamina. Mouse model carrying H22B lamina mutation develop muscular dystrophy and dilated cardiomyopathy, similar to human striated muscle laminopathies. Laminate H22B missense variant was identified in a family with autosomal dominant Emery-Dreifuss muscle dystrophy and cardiomyopathy. This article was published in Human Genetics, and in this article we found that there is degenerative muscle fibers were observed at the laminopathy model with central nucleation. Moreover, the ATPase function was affected, and CC oxidase function also was impaired in the laminopathy model, and succinyl dehydrogenase also staining was affected and impaired in the laminopathy model. Interestingly, we found that LSMEN2 knockout improved the survival rate in lamine-related muscle dystrophy mouse model, but this data is preliminary at the moment. Also, we measured the muscle function. As the rescued knockout model of laminopathy, and we found improvement in the force, in the twitch force, tetanic force, and force frequency as well. And I performed IHC immunohistochemistry staining to detect the COX-1 and ATP5F1 mitochondrial marker, and I found also the signal was prominent at the periphery. However, as the rescued knockout model, it seems that the distribution of these mitochondria has been normalized. And also this has been quantified with imaging analysis, as you can see here. Today's summary: LSM-EM2 protein localized at the neuromuscular junction regulate vesicle traffic, and the malfunction of LSM-EM2 leads to dysregulation of vesicle transport and subsequently subsarcolemmal mitochondrial accumulation. LSM-EM2 knockout is viable, fertile, and didn't show any histological or functional abnormalities. In the future, it is worth testing to subject LSM-EM2 knockout to different disease models, such as Duchenne muscular dystrophy and amyotrophic lateral sclerosis, to reveal its pathological relevance. Suppression of LSM-EM2 using antisense oligonucleotide therapy could be a therapeutic choice for Lamine muscle dystrophies and other neuromuscular disorders. And finally, I should acknowledge and say thank you to all people and all the co-authors who helped me in this project, and without them, this project will not have been done. Thanks so much.
Thank you very much for this wonderful presentation, Dr. Iman. We are open for questions, for any questions for Dr. Iman right now.
First of all, congratulations for the acceptance of your paper in the really high impact journal. And my question is actually, so it's very interesting that, the laminopathy model mouse, the phenotype is improved by, cross-mating with your knockout mouse. So yes, it looks convincing, but why you focus on laminopathy first of all? I didn't get the point actually. The results may be convincing, but I wonder why you thought about the laminopathy.
Yes, I know this question is common and it's asked everywhere. Really, why we choose laminopathy model? It was a choice of Shintai Sensei, the director of our lab. This is not the answer. But we didn't choose only laminopathy. We did investigation on other models. So, but I cannot say its name right now. So it is not the only pathology we tested to the moment. We tested others, but there was no effect. And to the moment, laminopathy is the one who gave us a good result, or we can see a hope. We observed a nuclear localization of LSM-EM2, as I said before. However, this nuclear localization observed as the overexpression model, not as the endogenous. So it was, we may see, we didn't believe it could be true or not. But there is no published data about this protein, and there is no study. We can say that this is the first study in the world studying LSM-EM2 in skeletal muscle. No one knows where it is exactly. And the only report about it says that it could be present perinuclear signal in C2C12 medium. So also no one investigated it endogenous and in vivo. The availability of a proper antibody is not present. So we thought that there is a nuclear localization, but we cannot say to the scientific world there is a nuclear localization unless we are 100% confident about this. From this point, Shintai Sensei chose the laminopathy model. And it gives us a hope that there could be, in the future, there could be a therapy for this rare neuromuscular disorder. So that's why we choose the laminopathy model, not laminopathy as laminopathy. We choose so many diseases, but laminopathy was the good one that worked with us to the moment. In the future, now we are currently working on Duchenne muscle dystrophy, and right now scientists at Osaka University waiting for the result, what will happen to the model of DMD with LSMEN2 knockout background. To the moment, no one knows.
Great. So, I think the next one of the next steps you may want to do is using the human cells, human muscle cells. Actually, we do have mutant cells from the patients, and also we actually have published some papers regarding laminopathy, and in which we introduce mutants like this. Exactly the same mutation as well. So you may want to study human cell, because all the data were from model mouse, right? So there is some difference between the mouse and human. So
Yes.
you may want to study human muscle.
Yes, of course, this should be done in the future. Yes, yes.
Anyway, congratulations.
Arigato.
So next speaker is actually my colleague from my laboratory. So it is my pleasure to introduce Dr. Rui Shimazaki. He is my young colleague. Even though he was listed as professor in the program, and there was some misunderstanding. He is a postdoc in my laboratory, but even though he is very young, he is very enthusiastic, and of course he is very good. So I think he is already, he is an MD, and he's a board certified neurologist in Japan, and also he got PhD as well. So he's been in our laboratory. He dedicated himself for understanding the mechanism, especially the genetic aspect of the disease. And I guess he will be talking on the well, so first of all, our laboratory actually aims to do molecular study to understand the mechanism and also to develop the therapy for muscle disease. So it's a pure research. But my lab functions also as a referral center for muscle disease, and we are involved in the diagnosis of muscle disease, both in genetics and also muscle pathology. So we do all the screening analysis, but it's not always easy. Of course, many cases can be diagnosed, for example, Duchenne muscular dystrophy by MLP analysis and also sequencing analysis. But there are not so many, but there are patients who cannot be really diagnosed by simple or screening testing. I think he will be talking on this type of patients. Okay, stage is yours.
Okay, thank you. I'm Rui Shimazaki, not Ichimazaki. And I'm a researcher in Dr. Nishino's laboratory. And I was given the opportunity to present today thanks to an invitation from Dr. Russia. So I would like to express my gratitude to this honor. So no conflict of interest. So do you know this journal? This journal is official journal of World Muscle Society. And the World Muscle Society is an association for muscle disease experts. And annual meeting is held every year. And this year, meeting was held at Prague, and we exchanged opinion. Anyway, the illustration is a certain famous clinical sign. Do you know this sign? Yeah. Sorry? Yes, it's Gauer's sign. It's not so specific sign, but it's Gauss sign is often seen in DMD patient. Let's watch the movie. This is kindly this movie was kindly provided by Dr. Russia and Dr. Nishino. Okay. Notably, extending the knee didn't take not so much time, but raising his upper body took much time. Check again. Oh. Sorry. Because in early stage of DMD patient, quadriceps extending the knee relatively preserved, but gluteus maximus raising upper body become weak in early stage of DMD. As you know, DMD is X-linked form disease and the most common among the muscular dystrophy. Its prevalence 4.78 per 100,000 males. Causative gene is DMD gene. Typically, they don't any symptom at birth, but from 1 year old, they show high CK. From around 3-year-old, they show some manifestation like running slowly or being unable to jump. From around 10-year-old, they lose ambulation. From 50-year-old, they show respiratory failure. From 20-year-old, they show heart failure. Some patients with DMD variants show milder form, and it is called Becker muscular dystrophy. Becker muscular dystrophy patient typically showed had onset around 7-year-old, and the symptom is milder. Ambulation is kept over 15-year-old. This is immunohistochemistry findings of the dystrophin. Dystrophin is encoded by DMD gene. In control, dystrophin expression is seen on sarcolemma, but in DMD patient, no expression of dystrophin is seen. But BMD patients, in BMD patient, faint expression of dystrophin is seen. So it's result in the difference in symptom between the DMD and the BMD patient. So why does difference occur? Let's review the translation. DNA is composed by 4 different type of nucleotides, and the bases are grouped into the set of 3 nucleotides to specify the amino acid, and this 3 nucleotide group is called codon. DNA is transcribed into the RNA precursor, and introns are spliced out, connected exon, and the formation of the messenger RNA. And messenger RNA serves the template of the protein. But DMD patient, exon deletion causes frame shift, and this prevents production of dystrophin. In BMD patient, exon deletion also occur, but frame shift, reading frame remains intact. So it's allowed for short but functional dystrophin. So how do we diagnose the DMD? These are the methods for diagnosing the DMD. Of course, the accessibility to these the genetic tests must be considered depending on region. But in Japan, we conduct the genetic test as along with this flow. First step, if we suspect a DMD or BMD patient, we will apply MLPA. MLPA is a method to detect the exon deletion or replication. Specific probe pairs are hybridized as DNA sample, and the pair of the probes that are perfectly hybridized are ligated, and it become amplifiable DNA fragment, and amplified using the PCR technique, and presence of exon detected based on the intensity of signal. If there is exon deletion, probes cannot hybridize, and there will be no signal. 70% of DMD BMD patient have exon deletion or duplication. So we can diagnose 70% of patients by MLPA. If we cannot detect the exon deletion, next we will apply sequencing analysis. Sequencing analysis is a method for reading nucleotide sequencing. Because DMD is very large protein, so small variant like missense variant has a minimal impact. But the variant introduced the nonsense variant. The protein cannot be created. Or if one or two nucleotides deleted or inserted, the reading frame shifts, and it becomes a premature codon, stop codon. Nonsense or small insertion deletion account for 29% of cases as a cause of the pathogenicity. So how do we diagnose the remaining 1% of cases? We will perform mass biopsy, and the mass biopsy findings, mass pathology finding indicate BMD or DMD. RNA sequencing will be applied. RNA sequencing is a technique that analyzes not only sequencing of transcripts, but also their quantity. RNA sequencing detects abnormal splicing events such as pseudoexon generation, exon skipping, or intron retention. This slide emphasizes the splicing process. DNA is transcribed into the RNA precursor, and RNA precursor undergoes splicing and form the messenger RNA and translate into a protein. Most intonic variants that don't affect, but in their case, under specific condition, pseudoexon is generated within the intron, and pseudoexon inserted between the exon and exon. This is a Sashin plot. Sashin plot figure. Sashin plot is a tool to visualize the splicing events. Upper figure is control one, and lower figure is the patient's figure. And control one splicing occur and connect exon 50 to exon 51. But in patient, pseudoexon generated, and half of the leads include the pseudoexon. The Sashin plot is Sashin plot use raw data, and the exon density look like sashimi, so that's why it is called Sashin plot. We applied RNA-seq to all undiagnosed DMD cases, and we detected splicing abnormality in all cases. This means technically all cases are diagnosable, but why do we insist on achieving genetic diagnosis? Because DMD is a treatable disease. So I would like to talk about gene therapy. In Japan, Vitrolsen has been released. This drug works by skipping exon 53 and remain to restore the reading frame. In other words, the drug aims to convert DMD patient into a BMD patient. The other drug, it introduced the genes encoded the mini dystrophin. In addition, the treatment is being developed to skip the pseudoexon. So personalized treatment will be available. So therefore, definitive genetic diagnosis is important. This is my last slide. DMD variant is although accessibility to genetic tests should be considered, but DMD variant is technically diagnosable in all cases. Genetic diagnosis is crucially important because therapy tailored to specific variants are possible. Thank you for your attention.
We do have time for questions. So maybe I'll start with a question. There's obviously a lot of therapeutic development in the field of DMD. What you have shown, the antisense oligonucleotide-based therapies, but also the mini gene therapies to introduce a small copy of DMD. Maybe you can give an overview as to where we stand with the different therapies, like which ones have been approved, which ones are in the pipeline, and which ones you consider have the most potential, like for the treatment of your patients.
Sorry, you mean the which is the better or less side effect or something. So you ask about the which is
yeah, so maybe you can discuss kind of the therapeutic potential of the antisense oligonucleotide therapies versus the mini gene or gene therapy therapies for DMD.
Yes, but exon skipping therapy is not complete therapy because the therapy aims to convert DMD into the BMD, but mini gene therapy is provides a short dystrophin. But I heard mini gene therapy has a very rare, but has a severe side effect. So both are good point or bad point.
Thank you very much. Thank you, Dr. Rui. Following this question, actually, as we did give one patient, Egyptian patient, the exon skipping therapy, yes, exon skipping therapy just changed the patient from being Duchenne to Becker, which is better than nothing, but especially if we start as early as possible without losing the muscles. Although the gene therapy, actually it changes the problem from the start. I mean, in all the cells. So changing the gene in all the muscle cells will improve the patient, especially if we take the gene therapy as early as possible, like when the muscles are still there. I hope, I did not experience this yet with any of my patients, but I hope that in the future we can do that. The FDA approved this gene microdystrophin gene therapy last year only for patients with age group from 4 to 5, or I think, yes, 4 to 5 years. But this year, actually, it was approved for all age groups, and it was approved for ambulant and non-ambulant patients, and I think this is like a milestone in the journey of treating Duchenne muscular dystrophy, but still we don't know how the effect of this gene therapies will be on the future, especially with the in-ambulant patients, how the muscle cells will improve, or will the muscle cells will regain its power or not. So actually, yes, as Dr. Rui mentioned, the diagnosis procedures that he mentioned is very important for us, which will lead us to better choosing of gene therapies, whatever is it, like gene modifying or gene therapies. Thank you.
Any other questions?
So I want to add some comments. So the gene therapy is applicable to any mutation. So that is one thing you can consider. But at the moment, the biggest problem is, again, this is not full-length dystrophin. Dystrophin is very, very big. So it cannot be accommodated in the vector. That is the problem. But actually, the people, are, they are trying to develop, kind of full, dystrophin gene therapy. Of course, it's not available yet, but they try to use 2 vectors and combine together. So this may become available in the future. So as I mentioned, this field is really developing, under development, yes. So will be changed, yes, in the future.
Is there any way that they try to to correct the deletions present in the exons using CRISPR technology or something like that?
That is also another, actually, the theoretical approach. But so far, clinically, I mean, in terms of model mouse, it worked. But in human patients, it's not really worked, not really worked yet.
Excuse me, I can see there's a great interest in gene therapy for children with Duchenne muscular dystrophy, but I can also notice you all agree there may be some delay in selecting the appropriate patient that could be candidate for gene therapy. From your perspectives as an international expertise in Duchenne muscular dystrophy management, what can be other proactive mechanisms that can help us to save muscles from wasting or atrophy until we decide or until we choose appropriate gene therapy for these children? Thank you.
Because I'm a clinician, so I had to jump in. So as you know, that the only approved treatment for DMD is deflazacort, which was approved by the FDA a couple of years ago to help stop the muscle from deterioration because of it's an anti-inflammatory process. And it's better than the steroids as regards the side effects. But please make sure that you give your patient all the right advice about the nutrition and about the side effects of such medications, so not to reach this point, and we have to stop the treatment at this point. So nutrition is very important. Physiotherapy is really important. It keeps the muscle going and avoid the contractures, avoid the scoliosis. So you prolong the ambulation of your patient. And if you prolong the ambulation of your patients, you will stop the respiratory muscle affection. You will avoid many psychological aspects in your patient. And when you start maybe exon skipping or gene therapy, then the muscle will be still there to give you a good, okay, thank you.
Okay, I think if there are no additional questions, we would like to thank you again for the excellent talk and also for the great discussion. Thank you.
At the end, I would like to thank our international experts and all the attendees who came from many cities in Egypt just to attend our DMD course, educational course. Actually, I benefited a lot from the discussions, and please remember that tomorrow and the 3 coming days, we will have the course in the hospital, in NG Community Hospital, where Dr. Linda Lewis will give a long day of teaching about the functional assessment of DMD. And if we're going to work on research for DMD, we don't want to forget clinical research, like understanding how to assess the DMD patients is very important for us. Natural history study is very important. We don't have any studies coming out of Egypt about natural history study, which is actually the important point to start for reaching therapy for our patients. Thank you very much.
Thank you everyone for being a part of this important journey. I'd like to inform you that lunch is now right outside the hall, and thank you so much for attending today.